This protocol can be used to optimize both protein and primary antibody concentrations in a single assay plate and to perform 2 assays using a single sample preparation. The main advantage of this technique is that it allows 2.5 days of lab work, including data analysis, to be completed in 3.5 hours.
This technique provides a high-throughput assay format for developing a blood-based biomarker for ALS and is an ideal method for conducting large clinical trials. This method can provide insight into target protein profiles during disease prognosis and can facilitate the monitoring of the effects of drugs of interest in clinical trials.
After collecting 100 microliters of human platelet lysates from ALS patients and healthy subjects, fill out in-house generated templates for the capillary layout and sample preparation. The sample mixture preparation table is dynamic and will automatically calculate how much volume should be removed from the source. Place all the assay reagents on ice, except the standard pack, which should remain at room temperature. To prepare the fluorescent master mix, add 40 microliters of deionized water to a clear tube of DTT. And add 20 microliters of 10x sample buffer